compounded by the misuse of antibiotics and the presence of resistance reservoirs in different environments, antimicrobial resistance, or AMR, stands as one of the most urgent global health challenges of the 21st century. Addressing this crisis is further complicated by a lack of global urgency, fragmented sectors, and broken innovation networks that make the path to effective solutions even more difficult. This is especially relevant to countries like South Africa, where food and waterborne surveillance and reporting systems are few to non-existent. Here at the Department of Plant and Soil Sciences Food Safety Research Group, based at the University of Pretoria, we have focused our research on surveillance of foodborne and AMR pathogens within agricultural environments. Published earlier this year, a comprehensive report by our group consolidated findings from studies conducted between 2016 to 2019. The research focused on antimicrobial resistant E. coli isolated throughout the farm-to-fork chain in South Africa, offering critical insights into the spread and impact of AMR within this context. Obtained from various environmental sources, such as irrigation water sources and various fresh produce types, the 65 E. coli isolates included in the study were characterized using whole genome sequencing. These included irrigation water samples from boreholes, rivers, and dams, as well as fresh produce, including tomatoes, lettuce, cucumbers, and spinach, collected at various stages along the food chain, during harvest at the farm level, through transportation, and at points of sale such as markets, grocery stores, and schools. From a food safety perspective, identifying these microbial contaminants throughout the food chain is vital for hazard characterization and protecting the public. In summary, over 95% of the bacteria studied were resistant to multiple antibiotics, including 3 or more drug classes often used to treat infections. The bacteria also carried numerous genes that helped them resist medicines, while also aiding them in spreading between other bacterial species. Additionally, the isolates also had traits that make them more likely to cause disease in animals and humans, while some were identified as high-risk strains that could lead to infections if they contaminate our food and water supplies. This research matters as it shows that our environment can be a hidden reservoir of antibiotic-resistant bacteria, which are harder to treat and can spread through food chains. These bacteria might not make you sick immediately, but they can carry resistance to critical antibiotics, making future infections harder to treat. Studies like this are important as they provide vital information and insights about the quality and presence of potential pathogens within our irrigation water and produce, advocating for more regular water and food testing to catch these harmful bacteria early. Future studies should focus on developing innovating control and mitigation strategies to curb the AMR crisis within the environment. This feeds into the One Health approach, which recognizes the interconnectedness between humans, animals, and the environment to curb the AMR crisis.